Okay, let's talk about the important steps or events that occur during mitosis. Okay, so we are talking about mitosis, which is basically asexual type of cell division. Asexual cell division. When do you need mitosis? When you are repairing your broken cells or hurt cells, or when you are essentially growing new mass of cells. Another term that is used to refer mitosis is cloning. Why it is called cloning is because it produces identical daughter cells. I'm sure you have watched the movies, the Star Wars movie, something about the clones. Okay, so that essentially, remember in that Star Wars movie, all the clones are exactly identical, correct? So here, that is what I'm saying. In cloning, you produce the next generation which is identical, okay? In mitosis, that is what happens. And why is it important to have identical daughter cells? Imagine you yourself getting hurt on the skin, okay, or on your arm. You want the exact same cells that were there before you got hurt, right? You don't want any different cells. So if you want the same exact identical cells, you want to heal your wound by mitosis type of cell division, okay? You do not want some different patch of skin coming up. That is why mitosis in repairing or in growing, when you're growing from baby to adult, okay, it's also called as cloning. Why? Because you're producing identical daughter cells. Now let's take a look at the events that occur during mitosis, okay? The events of mitosis are basically 4 of them. In your textbook, it gives you 5, but I would I would highly encourage you to just remember 4 basic steps. It is called as PMAT: prophase, metaphase, anaphase, telophase. Okay, so essentially I am going to club together the prophase and the prometaphase into the prophase. Okay, so prophase, prometaphase, that is given in your textbook, club that together into prophase, and there are only 4 events that really happen during mitosis. So let's take a look at what really is happening during mitosis. For our understanding, I'm going to take an imaginary cell which has only 2 chromosomes. Just for our simplicity, it has this one red chromosome and the other one a green chromosome. Okay, so what is the 2n number of this cell? The 2n is 2. This is a diploid cell, okay, which has one red chromosome, and its homologous chromosome is the green chromosome. Okay, so this is the chromosome, or this is the cell, okay, that we are going to talk about. Now, essentially, what happens during prophase is that before even you come to prophase, there is interphase. So before prophase, there is interphase. In interphase, during S phase, the chromosome gets duplicated. Okay. Now here is what is happening. At prophase, your nucleus, your DNA is going to get fragmented, and chromosomes will be formed in the prophase. Chromosomes are formed. Second, the nuclear envelope disappears. So these are the 2 events that really occur during the prophase of the mitosis. Okay, so you start seeing chromosomes for the first time in the cell's life cycle. Okay, from prophase we come to metaphase. So what really happened in prophase? You started seeing chromosome, or chromosomes really appear, or chromosomes are formed. And second, the nuclear envelope disappears. These are the two events for your prophase. Coming to metaphase. In metaphase, what happens is the cell now becomes a little bit like an egg. It gets elongated on both the ends, and the two chromosomes that are present in the cell, they come to lie at the equatorial plate, an imaginary equatorial plate of the cell. Okay, and they are both attached onto the two poles of the cell. They are attached by the protein, and these are called as spindle fibers. Okay, and this is called mitotic spindle, and the imaginary the plate is called metaphase plate or equatorial plate. So what happens during metaphase? In metaphase, the chromosomes align at the equator. Okay, now that's what is happening in metaphase. From metaphase, the cell enters into anaphase. Okay, what happens at anaphase is, okay, from these poles, okay, the protein fibers are going to get pulled back. Okay, so what is essentially happening is the length of these protein fibers or the spindle fibers reduces, and attached to that comes back your chromosome. And now, since the chromosome has two arms, it basically gets split at the center. So one V comes to one side, the other goes to other side. Same thing happens to the other green chromosome. Okay, so essentially the chromatids are going to get pulled apart. And what are these two arms of the chromosome called? They are called the sister chromatids. So what happens at anaphase is that sister chromatids get separated. Okay, now from anaphase we will go into the last phase, which is called as telophase. Usually telophase immediately follows another phase called cytokinesis. Cytokinesis is essentially splitting of cytoplasm. That is why the term is called cytokinesis. It essentially means splitting of the cytoplasm. And at telophase, what are you going to see is the cell will start splitting from the middle, and the two daughter cells will get the equal amount of DNA and rest of the material. And as the cytokinesis progresses, the cell membrane between the two gets darkened, forms completely, and now it basically results into two independent daughter cells which have the same number of chromosomes. So what is going to happen at telophase? At telophase slash cytokinesis, two daughter cells are getting formed. the nuclear envelope starts to reappear. Comes back or reappears. And instead of chromosomes, now you are going to see DNA in the nucleus. Okay? So essentially, telophase is exactly opposite to your prophase. The events that occur during the telophase are exactly opposite to the events that are occurring in your prophase. Okay. Now before that we were talking about interphase. Okay, so let me just come to that first. So before the prophase, there is what is called as interphase. A cell which is present in the interphase essentially only has half the chromosome, not half the number. Okay, it has only single chromatid. Okay, so from here it enters into the prophase. Okay, in the interphase it is going to get, the DNA is going to get replicated, going to produce two arms for each chromosome. See here there is just single V, here there is inverted V. Okay, that is happening because the DNA gets replicated. That is possible because of replication of the DNA. So essentially this is your parental cell. Okay, now parental cell has 2 chromosomes. The number of chromosomes that you are starting with is 2. Okay, it gets divided, it goes from prophase to metaphase. Metaphase, which is marked by the equatorial plate. Anaphase, which is marked by the separation of the sister chromatids. And telophase, which is marked by the reformation of the 2 new daughter cells. So okay, at the end of mitosis, every daughter cell, if you take a look here, and now if you were to compare it with the parental cell, it is exactly identical. The 2 daughter cells that are formed, okay, the 2 daughter cells that are formed at the end of mitosis carry same number of chromosomes. That is really, really important. Okay, so remember that the 2 daughter cells have same number of chromosome just like its parent. Okay, and that is mitosis for you. And that is all for today, and we'll talk about meiosis in the next class.